So now that you have a little bit of context and background for the pentose phosphate pathway, let's go ahead and jump into it. Now, this pathway, like I said previously, is a little bit different from your kind of linear pathway of glycolysis, in part because it has some kind of like a cyclic component to it. When looking at this pathway, I think that it's very easy to become overwhelmed, in part because if you just were to follow it, starting here in the upper left-hand corner, and follow the arrows, you'll find that you not only you follow kind of a straight left to right path, but then you split off, which is inherently a little bit confusing. How do we go from one molecule to two different molecules? And then if you keep following those arrows, sure, they do follow a little bit of pathway, but then you kind of end up back where you started. Now, that's something that is, to me, inherently confusing about the pentose phosphate pathway. But I assure you, it's something that is easily processed. Now, before we go any further, what I'd like to point out is our starting molecule in the pentose phosphate pathway is our old friend glucose 6-phosphate. Where did we get glucose 6-phosphate, and where do we talk about glucose 6-phosphate? We talked about it in the context of glycolysis. So we talked about this molecule resulting from the activity of, I'm going to label it HK, hexokinase, taking glucose and phosphorylating it at position number 6 on carbon number 6, adding a phosphate group to that, making glucose 6-phosphate. So if you remember, one of the things that we talk about with glycolysis is we talk about the first or a key regulatory step, or the first regulated step, and the first committed step. Well, hexokinase is a first regulatory step. It's an irreversible enzyme that is going to generate a molecule that is not destined specifically for oxidation into pyruvate. Instead, what we see is that glucose 6-phosphate can go a couple of different directions. Immediately here, we start the pentose phosphate pathway with glucose 6-phosphate. Now, the next thing that's important to recognize about the pentose phosphate pathway is this right here. Now, for context, this is basically the top portion of our molecule, of our pathway. Now, what I think is a very key part of your understanding of the pentose phosphate pathway is looking right here. If you look at each one of the places where I'm putting a little plus or a star, what I want you to recognize is that you see not one, not two, but you see a total of 3 lines going towards an arrow. And that is not some stylistic implementation or some stylistic design idea. That is for good reason. The reason being is that when we start the pentose phosphate pathway, we have 3 glucose 6-phosphate molecules that enter the pentose phosphate pathway. So there are 3 simultaneous reactions catalyzed by glucose 6-phosphate dehydrogenase to make a total of 3 6-phosphoglucone delta lactone molecules, 3 6-phosphogluconate, and finally we end up with a total of 3 ribulose 5-phosphate molecules. Okay, so we're doing this 3 times. What that means is that every single place where we see NADPH, one of our products for the pentose phosphate pathway, we're actually getting 3 of those. Here's a second one, or our second one. You've got 3 of those. So when we start the pentose phosphate pathway, we've got 3 glucose 6-phosphate molecules. We generate a total of 3 ribulose 5-phosphate molecules. Now, what's important to recognize about these steps is that, well, basically what's happening, this top portion of our metabolic pathway displayed here, this top portion is what we're focusing in on initially, okay? Now, that top portion is somewhat commonly known as the oxidative phase of the pentose phosphate pathway. The reason for that is that glucose 6-phosphate is being oxidized into ribose or ribulose 5-phosphate. In the process, we're generating a total of 6 NADH, or NADPH, I'm sorry. We're also generating a total of 3 protons and using 3 H2O molecules. Now, at each one of these steps, step 1, what we're doing, or the type of reaction that we're catalyzing, is an oxidative or oxidoreductase reaction. Step 2 here, we're using water to open up this ring. So we're using water to break a bond. We're doing a hydrolysis reaction. Our third and final step in this oxidative phase, what we're doing is we're removing this CO2, and therefore we're going from a 6-carbon molecule, 6-phosphogluconate, to a 5-carbon molecule, ribulose 5-phosphate. So we're removing 1 carbon, and we're generating some more of our last step of our NADPH. The first step in this pathway are glucose 6-phosphate dehydrogenase enzyme. What it is doing is it's taking our glucose 6-phosphate and producing a lactone. So rather than having this right here, this OH and our hydrogen on carbon number 1, instead what we're going to get is a double bonded oxygen, giving us a molecule that is a lactone. So that's catalyzed by an oxidoreductase, which I'm going to label it as a redox reaction. So we still have our ring intact there. The next step is to open up that ring, done by our lactonase, and our final step is to remove that CO2, done by our dehydrogenase. Now, this first step is actually a pretty important step from, I would say, a medicinal perspective. So this glucose 6-phosphate dehydrogenase, targeting in here, this is actually a very important enzyme, fairly well studied. The reason that it's fairly well studied is that approximately 400 million people suffer from a glucose 6-phosphate dehydrogenase deficiency. Now, in general, this deficiency does not cause a significant problem and can be managed with dietary adjustments and considerations. So what this does lead to, however, is it can lead to it can lead to an increased concentration of peroxides, those reactive oxygen species, which the increased concentration of peroxides can cause lipid and protein damage. So lipid damage, well, potentially damaging some of your membranes. Protein damage, potentially damaging membrane proteins, but also maybe some cytosolic proteins. Now that can lead to a condition known as hemolytic anemia, where your red blood cells burst. And that hemolytic anemia can be triggered by a number of different things, including aspirin, by taking too much aspirin or maybe taking it too frequently, certain antibiotics, and some of these antibiotics are fairly common. For instance, nitrofurantoin is an antibiotic that can trigger hemolytic anemia in someone with glucose 6-phosphate dehydrogenase deficiency. And that's an antibiotic that is pretty commonly used for urinary tract infections. You can also be susceptible to hemolytic anemia by taking certain sulfonamides, which are generally used to treat bacterial infections. Now, in the event that you have a glucose 6-phosphate dehydrogenase deficiency, it's probably something that, well, it's definitely genetic. And so it's possible that if you're afflicted or if you have an affliction to this, then through your cousin or your grandfather or something who knows that. But these are definitely things that you want to be aware of, so that in the event that you're admitted to the hospital and you can't communicate, well, hopefully your medical records show that yes, you have this deficiency. Other things that can trigger this: fava beans as a part of your diet, and yep, you guessed it, mothballs. So some of these things are, well, mothballs are fairly easily avoidable, I would imagine. and fava beans as well. But antibiotics, that's definitely something that you would probably have had a response to and would be well aware of. At any rate, this reflects, and I'm going to jump back here. What I wanted to show here was the oxidative phase of the pentose phosphate pathway, oxidizing glucose 6-phosphate down to ribulose 5-phosphate, taking a 6-carbon molecule and removing one of those carbons. How do we do that? Through a redox reaction, a hydrolysis reaction, and a subsequent redox reaction. So redox, hydrolysis, redox. And in the process, we remove a carbon dioxide molecule. All right. Well, I hope this was helpful as you look at the oxidative phase and you look at this entire pathway as a whole. Remembering that you have 3 identical glucose 6-phosphate molecules going through this. I like to think about this as kind of a total amount. You started with 18 carbons in the oxidative phase, you end with 15 carbons. And if part of our goal in metabolism is to take a complex molecule and break it down as much as possible, like what we've talked about with taking glucose and breaking it down to pyruvate, I think that's a very good perspective to have. So what we've done is taken 3 6-carbon molecules, and now we've ended up with 3 5-carbon molecules, releasing 3 CO2s in the process. Well, I hope this is helpful, and have a good one.